Today on Metascience Matters, I'm speaking with Florian Naudet, Professor of Therapeutics at the University of Rennes. We discuss his work bridging metascience and clinical trials, whether esketamine is effective for depression, and how you can analyze data in different ways using vibration of effects analysis applied specifically to how we treat alcohol use disorder. This was a very fun conversation. If you'd like to support this podcast, please send it to a friend. And now, here's my conversation with Florian Naudet. You work on a wide variety of topics as a professor and a psychiatrist. So I would love to start with your origin story. How exactly did you become interested in psychiatry? How did you become interested in metascience? And how did you end up merging and blending the two in your work?
Yeah, so that's a good question. Actually, first of all, I was like many students in medicine, and I wanted to be a general practitioner. That was my original idea. But then I made some experiences in psychiatry. I went in a very specific unit, and I saw what psychiatry was about. And I found in psychiatry there was many ideas. You have folks involved in psychotherapies. You have folks involved in psychopharmacology. Other people are more involved in humanities, in neurosciences. And actually, I found this super diverse and super interesting. I found it that it was a very interesting field for research. And then I decided to do a little bit more of research, and I found that it was a perfect place to do some research. So I decided to go in psychiatry, but with the idea to do research at the same time. And that's why I came to psychiatry.
Wow, very interesting. And were there any, in terms of clinical phenomena, were there certain conditions or diseases or other things that drew you to psychiatry?
Yes, for sure, you have some fascinating conditions that many people don't understand, and most people don't understand. I believe I don't understand most of these conditions in terms of what's happening, and I found this very fascinating as an area of research. But also, I must say that I found that in psychiatry, I found many colleagues that were very interesting. I found a structure of clinical activities that was a little bit less hierarchical than the hierarchy we are used to see in medicine, in many specialties, with doctors that appeared to be more simple, a little bit more kind. So I believe that it was a good way to work with people who could be kind enough or in my style. So I went there also for that. But to be honest, later I figured that there was a lot of hierarchy in psychiatry too.
Can you elaborate more on that? What is the hierarchy within psychiatry that you experienced?
No, you have this kind of hierarchy between people who are so-called experts and people who are so-called clinicians in day-to-day activity. And those kind of experts often works in a university settings and believe like they know a little bit more than the others and have perhaps a little bit more power than the other too, uh, because of the status, like being a professor and so on. So of course now I have this status, but but I think this is very strong in medicine. This kind of hierarchy, very strong. So basically,
And
sorry.
throughout, sorry,
No,
go
this
ahead.
is something we see, for instance, in cardiology, in a lot of specialty, because you have also in medicine a lot of kind of hierarchy between different medical specialties. But actually, I found it in psychiatry too. And to be honest, I was wrong when I thought that there was no differences. But when you are making your choice, you are often very wrong. So you are making many choices based on wrong reasons.
And throughout your journey, moving through medicine and psychiatry, where did metascience come along? How did you become interested in
Yeah.
metascience?
So as I told you, I was interested in psychiatry because of the many diverse fields you can explore in psychiatry. So psychotherapy, neurosciences, humanities, and so on. And when I started to go in that direction, I didn't want to be focused on one topic. I didn't want to be the guy doing neurosciences. So basically, bringing my patient to the fMRI and doing some research on brain anatomy or brain function and so on. Uh, I wanted to have a broader view. So first of all, I didn't go directly in the field of meta research, but I went in the field of methods, and I trained as a methodologist. Well, I told you that many choices are for wrong reasons, but there was also another reason, because here in France, after your year 6 in medicine, after the year 6, you have to choose your specialty. So I chose psychiatry, but you have also to choose a town where you want to live and to do your activity. And many people choose Paris, especially when they want to do research, because in Paris you have a lot of possibilities. You have a lot of units, you have a lot of people, and it's much more easier to do your way in research. But we decided to go in Rennes, in the west of France, for the purpose of quality of life. You know, it's west of France is like west of America. It's west coast. So we have the sea, we have the so okay, we have the weather that is pretty bad, but except for the weather, I am in a town called Rennes, which is also about rain. It's always raining here, but except for the weather, here you have a fantastic quality of life. You can go to the seaside. You can. So we decided to go in Rennes. And then here in Rennes, I tried to see what could fit with my interest, and there was a master degrees about methods. So I said, oh, basically, it's a good option. I can try to go there. But then I had another problem because I wanted to train in methods in Rennes, but there was nobody focused on methods in psychiatric research in Rennes. We have people in methods, very generic uh, opinion on methods, but not specific on psychiatry. So I had a few, I would say, months being a little bit alone and asking myself what I was going to do. And I had this nice book of biostatistics, and I love these books. And it was written by a professor in Paris. Uh, he was a biostatistician, Bruno Falissard. And I love this book about biostatistics. And one day I discussed with a colleague, and this colleague told me, but you know, Bruno Falissard is a psychiatrist, but I did not know. So actually, I contacted him. I did my master degree in Rennes, but I went in Paris for all the training session and for working a little bit in research. So I learned a lot with Bruno Falissard in Paris. So he's a super cool guy. He's a mathematician, epidemiologist. He's a statistician. He's also a psychiatrist. And moreover, he's very kind and very brilliant. So this is my tip. Uh, if you want to go in research, you have to choose a topic that you love because you will have a lot of time working on this topic. But you should also work with people who are very kind because this is super important. Not to choose a very toxic person in order to mentor you. I was lucky because I didn't knew him before, but he was very kind and very brilliant. And it was perfect to learn with him, actually. So we started actually on my PhD and on my medical thesis to work on these issues. We work on the issue of studying antidepressant research, and we wanted to know how much the design we chose to design our clinical studies might impact the estimates we have in those studies. So beyond this, there was a question of placebo effects and so on. So we work on that. And for my first studies, because it was very difficult to collect the data, we decided to do a meta-analysis. So my first study ever was a meta-research study and exploring issues that would be relevant for clinicians. So actually, I get in meta-research like that. And then I had some interest in meta-research. I have some interest in placebo research. So after my PhD, I tried to apply to both, actually. I first applied on placebo research on a team in Boston, perhaps close to where you are working. But actually, they didn't want me. So my application was rejected. This is something that is so super frequent in academia to be rejected. All our papers are rejected, all applications are rejected. We are always rejected. So that's like that. And then I tried to apply in meta research also. And I tried to apply to the Meta Research Innovation Center at Stanford, Matrix. Uh, and here I was successful in my application. So I went there. So you have a lot of, I would say, randomness when you are going in your career as a researcher. And sometimes it's a good luck. And this time it was a very good luck. Because here in Stanford, I met a lot of colleagues super interested in meta research, always kind and brilliant. So very kind and very good people like Bruno Falissar was. So I met there John Ioannidis, I met there Stephen Goodman, I met there David Mower with from Canada. But you know, in Stanford, everybody wants to go in Stanford. At least at this time, I don't know, but a few years before, everybody wanted to go to Stanford. But I think it's always the same. And we discussed a lot of topics. I learned a lot there. And then I think I became very involved in meta research at this time.
Such a fascinating journey that you're on. So I would love to start by talking about one of your really fascinating studies that came out recently. Speaking of blending metascience and psychiatry, this study that you published a few months ago is not just a registered report, but it's a registered report of a systematic review on the efficacy of esketamine for treatment-resistant depression, summarizing a bunch of double-blind randomized controlled trials, which are often discussed as the gold standard in clinical kinds of studies. And this was also an individual patient data meta-analysis. So I would love for you to talk about how this study came about, what exactly is an individual patient data meta-analysis, and the questions you aim to answer and what you found.
Yeah. So as a um, I think this study it was very long to do and difficult to do. So you should not when you are conducting this kind of study, you should be aware that it will be long and much longer than you expected. Where does it come from? Actually, it comes from it comes. So I have to digress a bit. Sorry for that, but I will digress.
Please, we have
Yeah,
time. Take us on a
but
ride.
I told you why I choose psychiatry, why I choose methods, but also I get trained in clinical pharmacology and therapeutics. And during this training, I had to work as a methodologist, so as like an epidemiologist, helping my colleagues to design their own studies and so on. So meta research is like the basic science of methodologists who want to understand why they are choosing these methods. So with meta research, we are trying to explore the science of methods. But when you are a methodologist, you have to use those methods in the practice. And it's super important to do that because you learn also the dark side of clinical research. And I don't say that in a negative sense. I say that because when you are a meta researcher, it's super easy to criticize what you have found. So you can easily say that there is bias everywhere, that there is false positive results everywhere. So you can be in that kind of complaint with meta research. And that's fine because there is bias everywhere. There is all this stuff everywhere. It's fine to say that. But it's much more difficult to do research than to criticize research. So when you are doing research, you are learning a lot about what you are talking about when you are a meta researcher. And I was used to be a methodologist for many studies. And when I started, I worked on studies on treatment-resistant depression. And this is exactly the patient that we, so not exactly the same patient, but this is exactly the same entity that we are dealing with esketamine. But there is a difference. When I started in the field of treatment-resistant depression, we used this terminology that is not an established terminology in psychiatry, but we used it to select patients that have tried almost all and every treatment that exists on this earth. So basically, why we did that? Because it was for studies of very invasive treatments. So a few years ago, we tried treatments like deep brain stimulation. You know, the kind of stimulation you propose to patients with Parkinson disorder. So it involves neurosurgery. So it's super invasive. So because it's super invasive, you cannot propose this to a patient that could have another treatment that might help him. So we people at this time build the studies to select patients that had really no other option of treatments. So it was a very long list of selection criteria, of inclusion criteria or non-inclusion criteria. And for us doing research, it was super difficult to find the patient because the list was so long that it was very difficult to find a patient that has tried this treatment, this one, this one, this one, this one, this one. And basically, it was difficult to do these studies. And treatment-resistant depression was a terminology used just to describe that we have selected very, very severe patient not responding to anything in order to do those invasive studies. And the term resistance is not really a good term because they don't become resistant. It's not like the treatment works and then they get a resistance, as you can develop a resistance to antibiotic when you are a bacteria. It's not exactly the same. It just means people don't respond to a lot of drugs or to a lot of other treatments. But then with esketamine, this is super interesting because we are a few years after. We are maybe 10, 12 years after that. And now treatment resistance is becoming mainstream. And while it's becoming mainstream, people consider that those patients, do not respond to a lot of treatments. But the definitions now are very lax. It's 1 or 2 drugs that were tested before claiming you are resistant. So it enlarges the market share for this kind of treatment resistance. 10 years ago, it was very difficult to find the patient. Now, maybe 50% of the patients are resistant because the definition changed. And that's why it's super important because esketamine was marketed and get a market approval by the FDA or the European Medicine Agency for treatment-resistant depression. And this is super interesting because not really for esketamine. I'm not so interested in esketamine. So as you will see, I don't have a good opinion of this drug, and I think I'm not going to use it a lot in my clinical activity. But I am more interested in regulatory science. And what's happening in regulatory science is that we have much and much more approvals based on preliminary evidence and the idea of your condition as an unmet need. So it's very easy, for instance, in oncology, to say that you have an unmet need. For instance, you have patients who have tried many drugs and don't respond. And then to propose them, like in a compassionate way, to have a drug with less evidence, providing there is hope it works. And the hope it works rely on a mechanism of action. And this has led to many successful innovation in oncology, but also to many drugs that entered the market without being proven to be effective later. So this is in the field of oncology, and I'm not in that field, so I won't comment more on oncology. But it seems that the same is happening in psychiatry, and people are trying to go in these accelerated pathways by stating that they have a new unmet need, a new condition that is very, very difficult to treat. And treatment-resistant depression is this kind of category. And with this kind of category, the companies can ask for accelerated approval, and they can lower the bar for the evidence needed. And then for esketamine, we saw that the evidence was very low.
So the amount of evidence, because from what the little reading that I've done is that clinical studies of ketamine began in the late 90s or early 2000s. Do I have that right, roughly?
Yeah.
So just how much investigation has been done, period, clinically of ketamine?
Can you repeat the question?
Like, how much investigation has there been clinically on the effectiveness of ketamine? Like, regardless of how strong or weak the evidence is, how many studies, what is the magnitude of the amount of investigation that's been done to date?
I cannot tell you with precision in ketamine because in our systematic review and meta-analysis, we focused on esketamine, which is an enantiomer of ketamine. Uh, by enantiomer, I mean that ketamine is a racemic form of the drug, and uh, one form is like that, and the other is like that. So pretty the same. So it's visual, sorry, but it's an enantiomer. And enantiomers can have some specific properties from the pharmacological point of view. For instance, for esketamine, maybe it's easier to put it in an intranasal formulation, which is better than the invasive way to give it uh, just by infusion. But it's also a way to do what we call evergreen. You make a minor pharmacological innovation to a drug to get a specific approval for a drug different from the other, but the difference is very small. And the evidence we relied on for esketamine were about 10th study, or not so much for esketamine, but there is much more for ketamine. But much less, I would say, strong because those are, in general, early phase studies and not like the phase 3 studies we had for esketamine. And they were maybe a little bit more heterogeneous. They can be more heterogeneous, for instance, in terms of dosage, but they can be also heterogeneous in terms of comparator. So, for instance, they can be do with a placebo, or they can use an active placebo, like midazolam, for instance, which is a drug that is not used for its effect on depression, because midazolam is a benzodiazepine that works basically on anxiety and has a short-term efficacy, not a long-term efficacy. But it's used to create a state of sedation that might mimic the state of sedation you can have with ketamine. This is something that is done. I'm not sure that it works at 100%, but something that is right. So there is much more heterogeneity in the literature on ketamine. But to be honest, I didn't review this literature. I focused on esketamine because esketamine is the treatment with the approval. And as I told you, I'm super interested in regulatory science, much more than in drugs in general.
So for this enantiomer of ketamine, esketamine, across routes of administration, across dosages, across comparators, the take-home finding of this study is that the efficacy of esketamine might leave a lot to be desired. It might not be that effective.
Yes. We found a difference that was small, perhaps beyond clinical relevance, even if it's always very difficult to determine what can be called clinically relevant in this area, in psychiatric. It's always difficult on those scales. But for that, we use the threshold that was used in designing the trial, in computing the sample size, and it was very below this threshold. So actually, the effect is quite small. It's quite in the same area of magnitude than other drugs for depression, than other antidepressants, than psychotherapies, than all these things. I don't say that all the things have a big effect size. I would say that rather all the things have a quite small effect size. And esketamine has also the small effect size. But esketamine is given intranasal formulation. Esketamine should be given, for instance, here in France, we give it at the hospital. So it's much more expensive to use it, to give it. And also, esketamine has some kind of potential safety issues. It can increase your blood pressure. So it's not always good to have an increase in blood pressure, especially if you have some cardiovascular risk factors. But also, it can increase the risk of sedation and put you in a state that is a bad state that you won't like at all during the sessions. It's not for all your life, but it's during the session. But it can also create some kind of sedation. So it has some safety issues. It's more expensive and it does not do much more than the other. And this is assessed in 4-week studies, which is very short, 4 weeks as a follow-up period. Especially when you know that treatment-resistant depression is considered as a chronic condition. So we have something that we consider chronic, and we are studying it on a short term. I mean this for initiation trials, because then you have this kind of continuation trials that are basically people that are remitters or responders to esketamine, and you continue the drug or you stop it abruptly. And those continuation trials are not always very good because you can have effects that are really related with the drugs, but you can also have withdrawal effects when you stop a drug abruptly. And also you are working on a pool of patients that are selected because they are responder. So you are going to overestimate the effect size because you are already working on the good responders and not on all patients you will give the drug. So I prefer to focus on initiation trials, and those are very short term, and they don't show a big efficacy for esketamine.
That's such a fascinating point about the continuation trials. I could definitely see ending up in a winner's curse type of situation where when you bring this out into the clinic, that the magnitude of the effect size could shrink tremendously.
Exactly. And this is well known not only for esketamine, but you have some meta research on that show that continuation trials always overestimate effect size. But it's pretty logic that they overestimate effect size. I think they don't really overestimate effect size. They just assess a different estimand than what you want to try in initiation trial. And I'm much more interested in the estimand by initiation trial as a clinician.
That makes perfect sense. So this study was a registered report, and I would love to hear about the process behind that, because with a registered report, you have a stage of peer review before analysis and data collection and all that. So I would love to hear about how that went publishing this.
Yes, it went very well. Actually, it's my second registered report. So it's not the first time. But I would say it's my second published registered report because I have a third one that is not published and that will never be published. I will tell you that after. Because I will start by stating that it's a very good format and I really love this format. And to be honest, I think this meta-analysis, in my opinion, but I am biased, is very important because it can inform clinical care and so on and so on. And I think that this meta-analysis, we could have done it as many research activities and then submitted it to a journal in a regular way. And I believe, but I might be wrong, that many so-called high impact factor journal might have been very interested in that. But instead, we choose to use a journal that proposes registered reports, and BMC Medicine is one of the only clinical medicine journal that offer registered report. We don't have a lot of choice. So we went to BMC Medicine, not because of the impact factor, but because of the standard and because it offered registered report. So this is actually, I think, much more important than the standard, the impact factor that is put forward by many journals. So then we went there and we had to submit our protocol. Basically, this research project, it was funded by the French Ministry of Health. So we had first peer review process with the funder. So actually, the protocol we submitted to the journal, it was already in a good shape because we prepared it. It was also peer reviewed before by the funder and the expert of the funder and so on. So we had the protocol already written. And I would say, as many of people doing research and going to funders, because we all write protocols for funders. So we had everything. So it was super fast to put it in shape, to send it to BMC Medicine, and to wait for their feedback. And the feedback, it was pretty fast, I would say, 2 months, maybe 2 months. So it's fast, but it's not that fast, but it's fast. And that after 2 months, you have perhaps 1 or 2 rounds of peer review, and you agree on all the details of the protocol, all the things. So it's very good because you have feedback from the reviewers, and at this stage, they can really have an impact on your research question. And say, no, this is not super important. You should rather focus on that. For instance, we plan to assess the efficacy of esketamine at week 4 or at the end of treatment. But the reviewers say, no, but esketamine should be a drug that acts much before. So you should have an endpoint that is earlier than 4 weeks. So we added that during the peer review process. We said, okay. They said also, you should have like a boundary for clinical efficacy. What would be the minimal clinical difference that you want to use? So we decided and we defined it at this stage as a stage 1 peer review. And then we were able to do our research. So we got an in-principle acceptance, meaning that whatever the result of your study will be, they will publish the study. So this is very good against publication bias, because if my study is negative, I would say negative like that, negative, actually, it's the same. It's published, and the journal won't judge about this impact based on the result. So this is super important and interesting. So we could start the project, but then it took a lot of time. And here the problem was starting. So it was long to get the data sharing agreement signed by my institution, because many institutions are not aware about clinical trial data sharing and that are not so used to sign this kind of very, very complex technical regulatory document that you sign with. For us, it was a platform called YODA, the Yale Open Data Access project. So we had to sign that. So it took time to my institution. And then we had a change of statistician during the study, because the first statistician he changed his activity. So we have to change the statistician to basically to start not from scratch, but to start a lot on our study. Then the statistician had some other duties at the same time, working on other studies. So it took perhaps 3 years or 4 years to complete the study. And this is something that we did not anticipate it. We thought it would be much more rapid, much more fast than that. But we did it. We were able to complete it. And then we sent it for peer review. And the second stage of peer review was super simple. They just checked that we have done what we have promised. A- and then they accepted the study because, of course, we have done what we have promised.
You raise a really good point there about having the protocol from the submission to get funding for the submission of your stage one report. Because I think a lot of people are sort of afraid that stage one, the preparation for it and the peer review takes a long time. But everybody has to go through a round of review to get the money to do science. So that material is readily used for your stage one report.
Exactly. So for us, it was fast. In this case, in this case, um, I would say that having your protocol ready for the funder, having your protocol ready for application for people who are doing research on routinely collected data, they often have to present a protocol to get access to the data and so on. So we all have this protocol. And actually, we can be transparent and do a registered report that is peer reviewed, but we can also register it. So it's not necessary to do the registered report, but we can also register it. And many people doing research on observational studies and so on do not even register the protocol, even if they have one. This is silly. They have one and they don't register it because sometimes they fear that if they do that, they cannot change the protocol. And this is a fear. And they have two fears. This one and the other fear is to be scooped. So about being scooped, there is something that is simple. You can ask for an embargo. If you registered your protocol on the Open Science Framework, there is an embargo possible, so you won't be scooped. Second, in my experience, I have never been scooped, but many times people had the same ideas than me. So they didn't took my idea. They had exactly the same idea because, I am not a genius. Many people are not genius, and maybe many people are having good ideas at the same time. It's like that. So being scooped, it's not my biggest fear. But the second option, people fear that they cannot change anything in the protocol. And to be clear, in our register report, we had to make some changes. For instance, we defined certain outcomes like suicidal ideation in a way that it was not possible to construct with the data we had. So we had to make some changes, but we had to be transparent about the change we made, the time we made the change, and the reason why we made the change. So actually, registering your study, doing your registered report, doesn't kill all your degrees of freedom you have as a researcher, but it implies that you are transparent about those degrees of freedom.
And how long do you think, like, given the time investment upfront at stage one, do you think that saved time on the back end once stage two was completed because you already had in principle acceptance?
To be honest, I don't know. I don't have like systematic evidence on that, so I can just tell you about anecdotes. So for my two published registered reports, stage 2 was super fast. And also stage 1 was super fast. And as I told you, I have a third registered report uh, that will never be published because we were not able to complete the study. And part of this was that we did this as part of a PhD student thesis, which is 3 years in France. And in this registered report, we had, I would say, more than 1 year in stage 1 peer review. We have 1, 2 round of review, 3 round of reviews, always a lot of time. And actually, we lost a lot of time for that. And then it was also based on data sharing, and we also had to apply for the data. And as I told you, for esketamine, it was long. And for this other registered report, it has been long too. And even longer because it was during COVID. And during COVID, everything was not so well organized and so on. And it was very difficult to get access to data. And then we had no time to perform the research because in the time window of a PhD, it was super, super challenging. So this is something I want to say. I really love registered report. I think it's super important, but it's kind of challenging, especially if you don't have the time. But for research, we need time, of course. But sometimes in the current research environment we have, it's difficult. So for a PhD, you have to do your study in 3 years. You have to deliver papers during those 3 years. It can be challenging. So now I'm not sure I recommend to my PhD student to do registered reports, even if I like it and I am so happy to have done this, and I will do it again. But you have to take the pros and cons before doing that.
That makes a lot of sense. In another study that you've conducted, you performed a vibration of effects analysis to look at the impact of inclusion and exclusion criteria, as well as various other analytical choices for treatments for alcohol use disorder. And in this study, you performed over 9,200 analyses. Can you talk a little bit about how we normally treat alcohol use disorder and what you found in this study?
Yes. So this is a super long story again.
We love long
Yeah,
stories.
but this is actually it was the first work I supervised as a mentor. It was the work by a PhD student called Clément Palpacuer, and with Clément we so I started with antidepressant research, and then I fell down in alcohol after depression. I went into alcohol, as is often the case. And this is I love to tell this story because this is a nice anecdote. I was at this time uh, I would say chief resident. I don't know if you have this term in USA, chief resident.
We
You have?
do.
Okay. So I was chief resident, and when you have our chief resident, I think you have a lot of duties, a lot of work, clinical activity. You have a lot of it's it's very easy to go in burnout. And one day, I had this guy from the pharmaceutical industry that came to me and said, you are a psychiatrist. You are also involved in clinical pharmacology. So we have a new drug that's coming into the market. It's called nalmefene, and it's coming in the market, and it's very new. It's like a game changer because they always say it's a game changer. And this is a game changer because for long we used drugs to maintain abstinence. So you make sure that people stop alcohol, and you give a drug in order for them not to get back to alcohol and to start again to drink. So this was a previous way of treating patients. And with nalmefene, basically, the idea was to give the drug for people just to lower their consumption. So the idea is that with the drug, they will drink less. Okay, but basically this was a new indication, but nalmefene was not so a new drug. It was very close to a previous drug that was called naltrexone, that was given to maintain abstinence and had an approval for that. And it was also given off-label to reduce consumption. So nalmefene was the first one to have this indication, but actually it was not like the big innovation of the day. But this, at this time, I did not knew because I did not knew the field of alcohol dependence. And he came to me and said, well, I need somebody to present the drug to all clinicians in Rennes, my town. And I said no, because I'm not interested to work for a pharmaceutical industry, because I wanted to be independent since the beginning. And I said no. And then he said, oh, well, but we discussed this with your boss. And if you remember, I told you that there was a big hierarchy in medicine. And when you are a chief resident and somebody comes and say, it's not me who decide for, it's your boss, you often say yes. So I said, okay, I will do this because I don't want any problem, but don't pay me. And they didn't pay me at this time. So I did it for free. So it's totally crazy. And it was super interesting because I went in this good restaurant. So I told you in Rennes, I chose Rennes for quality of life. So we have very good restaurant. We have very good crêpes. We have very good galette, which is a very nice dish from France. And we went in this restaurant with all folks involved in psychiatry, all folks involved in drug addictions, in alcohol disorders, and we presented the drug. And basically, it was very fun because I presented the slides about the biology of alcohol dependence. And when I discussed this with the guy, I say, but my focus is on methods. It's not on, mechanism of action. So I don't exactly know I know nothing about biology of alcohol dependence. And the guy from the pharmaceutical industry told me, it's not a problem. We have prepared all the slides. So you just have to learn the slides. We have all the text. So you just have to learn the text. I said, but okay, but if there's some question, we will have to answer your question. Right, Randy, I am answering your question now. But he said, no, don't worry. Because we have all the questions written and we have all the responses because we did this in many other towns in France. So we get many other questions. So we know basically all the questions that they will ask. And then we have prepared the response. So you just have to learn the response and to respond. And that's all. So I was basically a big quack in doing that. And I have very nice slides about the physiology of alcohol. So I was able this day, I'm no longer able to do that, but to tell you exactly if you drink some alcohol one day, where it will act on your brain, on what area and what it will do. So it was fascinating. I had a lot of fMRI picture. As I told you, I had no interest in fMRI. So I had all this stuff. I say, oh, it's here. This is the exact place where the alcohol binds the receptor of your brain and then get you a little bit drunk. And so that's what happened. And for my colleague, I had another colleague who was involved in discussing the clinical efficacy of the drug, the clinical evidence. And they also prepared the slide for him. But the slides were like the slides that you have prepared by yourself. My slides were very perfect. And it was very clear that they were prepared by the laboratory. There was even the logo of the laboratory. And so it was very clear. But for the slides of my colleagues, it was like he prepared the slides in his garage, working on his computer and preparing the slide of his thought. It was not the case. But it was just to give the impression to the public that this information was from him and not from the laboratory. So actually, it was my first experience of ghostwriting, and my only experience of things that were ghostwritten, actually. And it was super interesting to see this from the inside. And then they gave me a lot of documents because they are fully transparent. They give you everything. They give you the slides, they prepare everything, but they also give you a big dossier. And in this dossier, you had all the files about nalmefene. You have all the files, the regulatory files, and so on. And as I told you, when you are a chief resident, you are very close to the burnout. So if you give me a dossier, I won't be able to read it because I have so many things to do that is not possible. And the dossier was very, very big. But a few months later, I had more time. And I say it was fascinating. We have a new drug for alcohol dependence, very, very new, very fantastic, a game changer. And I was eager to propose to do some kind of meta-analysis for master degree students. And I said, oh, we have the perfect topic for a network meta-analysis, just to compare all these drugs in alcohol dependence in this new indication. And before doing my network meta-analysis, I did my bibliography. So I went in all the dossiers they give me. And I just look at the dossier for the approval at the European Medicine Agency. And I went into details. And I saw that it was a big mess. The the approval was based on a posteriori subgroup analysis of the clinical trial data. the clinical trial data was not convincing enough. The effect was too small, with almost nothing. Just a small, very tiny reduction in alcohol consumption. While it's not a very big outcome, just reducing your consumption by 1 drink or 2, it's not what you expect. You expect to reduce risks related with that. Because the problem with alcohol is not that you drink. I'm happy to drink. The problem is that when you drink, you have problems. You can go to the hospital because you have a car crash. You can fight with your friend, so it's not good for your friendship. You can lose your work because you drink too much, even when you are at work. So these are the problems, and these are the clinical outcomes we want to see. But then just measure tiny reduction of consumption. And because this was too small, the health authorities say, no, we cannot rely on that. And then the pharmaceutical company provided new analysis based on subgroup of patients, but those were not planned in the protocol. They were done a posteriori with all the possibility of doing some kind of p-hacking and arcing and so on. And this was approved. So basically, we decided to go in this direction with Clément, who would be my first PhD student, and we decided to do a PhD, a full PhD, on this drug, nalmefene. And we started to do first just a regular meta-analysis, and we saw, as for esketamine, that the effect was very tiny and very small, as many actually drugs in many conditions, but small efficacy. And then we performed a network meta-analysis, and we found no difference between nalmefene and naltrexone, the other drugs. And this was super interesting because at the same time, Lundbeck, the manufacturer of nalmefene, provided an indirect comparison of nalmefene and naltrexone and found a difference. So we had a case of, discrepant result for meta-analysis, which is a big problem because meta-analysis is supposed to be reproducible synthesis of all the existing evidence. So we had synthesis of all the existing evidence that was not reproducible. So we tried to think about that. And then we decided to try to do it by simulating all the possible meta-analysis we can do based on all the possible PICOs we can say. So we wanted to explore all the degrees of freedom that are available in meta-analysis. Because we know that we have a lot of degrees of freedom as a researcher when we are doing observational research. But what we wanted to know was if it could be the same for meta-analysis. So we basically perform 9,000 meta-analysis, a little bit more, on comparing nalmefene and naltrexone. And we found, in some case, superiority on nalmefene, in some case, superiority of naltrexone. In most of the case, no difference between both. And then we had a world record of doing meta-analysis. But we tried to beat our own world record, so we did many other vibration of effect studies, including in acupuncture on head-to-head meta-analysis, acupuncture versus, let's say, no treatment, or placebo, or shame acupuncture. There is so much heterogeneity for acupuncture that it's so funny. We did vibration of effects for individual patient meta-analysis based on all the pooled estimates all the study you can pull together, because most often IPD meta-analysis are not always exhaustive because companies choose to pull one study with another one. So we basically explore that. And we also explore vibration of effect due to network geometry in network meta-analysis. And here we made at least 8 million meta-analysis in the same paper. So basically, we hit the world record of meta-analysis, and for long.
That's such a wild story that you told about the a posteriori subgroup analysis leading to the approval by a government health agency.
Exactly.
Um,
Yeah.
yeah. Did when you finished this study comparing nalmefene and naltrexone, did you send it to that same agency?
No, but because I think it's not useful, because I think when the agency made their decision, they were super transparent. And the decision was made They are very competent experts, and to be honest, as I told you, it's much easier to criticize research than to do research. And folks involved in this agency have a very, very difficult job, much easier than mine. Because me, I am looking at their decision, I criticize the decision. But them, they are taking the decision. And they are super transparent at the European Medicine Agency because they publish the report with all the evidence, and they also publish the divergent opinion. So some people in the committee had the same opinion than us. So or they were already aware of all this stuff. So sending us our findings would not be new to them because they are already aware of that. But to be honest, it's super difficult to judge after the results are known if a drug is worse or not. Because if you judge the drug is worse, you are going to bring to the market snake oil, maybe sometimes with adverse events. Nalmefene has some adverse events. It blocks, it reverses, for instance, if you have an opioid use disorder and you use drugs like buprenorphine, for instance, you use nalmefene, it can block the effect of buprenorphine, and it can put the people in a very bad stage. So it can have some adverse effect. It can induce delusion also, something like that, or not delusion, delirium. It can induce delirium, something like that. So it has some kind of adverse effect, but they are not so frequent. Okay. But if you accept the drug, you can bring to the market something that is not so not so big efficacy, but a few adverse events. So perhaps some clinician would find it could be worth a try. And basically, sometimes you have people who are happy with that too. But if they decide not to approve it, they can deprive patient with something that might be effective. So when you have to decide, it's super difficult. How can you decide a posteriori of the threshold for success? That's why, following all this stuff, we have decided to think about how we can do this. And that's here I think really registered reports might be important, because with registered reports, we could bring this to regulatory science. And we could bring this to the regulatory science and to define a priori what will be the threshold for success in terms of clinical significance. Might answer the problems with esketamine. In terms of kind of analysis we want to have, subgroup analysis or not. And this, we can discuss this a priori with patients, because it's always super tiny when you involve patients at the end, because patients, they want to have drugs that might work for them. And if you say, no, there is an effect that is perhaps small, we are not sure, there might be, oh, okay, it's worth a try. I want to try it. I want to try it. But if you say this before having the evidence, what is the threshold for you to get the drug? I'm not sure that they will have the same discussion and the same definition. So that's why we didn't send our observation and result back to the EMA. They were already aware, and it's way too late. All this should be a priori and not a posteriori.
I love this point you just made. This is such a profound point, not just bridging registered reports with regulatory science, but making the public part of the registered reports process to define the criterion for what a successful outcome looks like. And it reminds me of the fact that on grant review boards in the United States at the NIH, there's usually a member from the public who's not a scientist giving their input. So if you could bridge the development and approval of study methodology, bridge that with publication and the production of published science, and integrate the public's perspective and how it impacts them to this process, that would be, yeah, that's an incredible vision, gotta say.
Yes, but it's, very naive, and it will never happen.
Today it is. Final question I'd love to ask you. If there's a listener at home listening to this, maybe they're in public school or they're an undergraduate, and they're very interested by some of the things that you've talked about today, what would you recommend for this person? What resources or books or other things would you recommend that they take a look at?
Um, I have so many things to recommend that went in my journey. Uh, if they are fascinating by the things we discuss about drugs and so on, I have a book that I love, which is called Pharmageddon by David Ely. It's a very nice book, a little bit complex. So if they are very young, it could be a little bit complex, but I think it's very nice book. If they are more fascinated by research, and we didn't address this in details, but I love the issue of reproducibility, and I love working on reproducibility. But there is this paper by Marcus Munafo and his colleagues, which is called A Manifesto for Reproducible Science, which is a uh, perfect way to enter in this area of meta research. And if they are not interested in drugs nor in reproducible science, we have something for them because we published this year a super study on meringue Christmas tree, where we try to see if we improve the recipe of a meringue Christmas tree with a reporting guideline, it could lead to better meringue Christmas tree. And actually, if you look at this paper, you will have the solution. It's a little bit disappointing on what we can do with reporting guidelines. But it's a way we have developed to discuss this with a younger public, because I think this is the most important message. We can discuss these issues of scientific integrity, reproducibility, problems in a way that is very distressing, because people will end with the message that science is broken, science is bullshit, and so on. And I really like the term bullshit, of course, but but this would be so wrong, because science is super good thing that happens to all of us, and science is made of debate with pros and cons. And of course, in these debates, we are providing observations that we collect with various type of designs, approaches, and so on. And this is complex. This is not so easy to get the evidence. This is not so easy to reach a consensus between scientific. And it's pretty usual to do that. It's pretty normal. So we should not be afraid and communicating about this in a distressing way, but we should just embrace the complexity of all this stuff and, you know, talk about this positively. So meringue Christmas tree are perfect to be positive, especially in the Christmas season.
I think that's the perfect place to end. Florian, thank you so much for talking to me today.
Thank you so much, Randy. It was a true pleasure and an honor to be invited to speak with you. And it was super kind to invite me. Thank you so much.
That was my conversation with Florian Naudet. If you'd like to support this podcast, please send it to a friend, and I'll see you in the next one.